everyone, my name is Han. Today I'm going to present my proposed study on how acute restraint stress and blocking androgen receptor in hippocampus affect anxiety, like behavior in male rats. Androgen receptors are the proteins through which androgen act inside cells. They are found in many parts of the body and brain, but we are focusing specifically on the hippocampus. Weinberger and Coglies found that androgen receptor mRNA is highly expressed in the human hippocampus, which suggests that the hippocampus is an important target for an androgen action. And we know the hippocampus is involved in emotion regulation and stress response. So androgen receptor signaling there might play a role in anxiety. Perseverance animal work suppose this idea. For example, Sulaga and Coglies found that male rats with impaired androgen receptor function show more anxiety-related behavior after mild stress and had a stronger corticosteroid response. That stress normal androgen receptor function might help regulate both behavioral and physical physiological response to stress. The most relevant study is Edinger and Frian's 206 study. They gave flutamide, which is an androgen receptor antagonist, indirectly into the hippocampus of male rats and found that blocking this receptor increased anxiety-like behavior. So it seems that hippocampus androgen receptor signaling might normal have an androgen reducing or anxiolytic effect. However, it is still unclear whether hippocampus and androgen receptor signaling has a general anxiety reducing role or become particularly important during acute stress. To address this question, this study aims to examine the independent and interactive effect of acute restraint stress and hippocampal androgen receptor blockade on anxiety-like behavior in male rats. I have two independent variables. The first is stress condition with two levels, acute trained stress or no stress. The second is treatment, also with two levels, flutamide or vehicle, and the vehicle is the same solution without the active drug. I predict that both acute restraint stress and hippocampal flutamide treatment will independently increase anxiety-like behavior. More importantly, I predict a stress by treatment interaction, such as flutamide will produce a greater anxiogenic effect in stress rats than in non-stress rats. Moving to the methylation, the will use 40 adult male long Evans rats. They will randomly assess to four groups, one with 10 rats in each group, and the house under standard laboratory condition with free access to food and water. As shown here, the study used 2 by 2 between subject design. These four groups allow us to test the mean effect of stress, the mean effect of treatment, and the interaction between them. This is the interaction expression produce procedure first. The rats will have blind guide cannulae implanted with the tips positions in the dorsal hippocampus. After surgery, they have about 7 to 10 days to recover. This guide cannulae allow us to give flutamide of vehicle direct into both sides of the hippocampus. After recovery, rats will receive bilateral interhippocampus injection of their fragment or an equal fragment volume of vehicle. Then two groups will undergo 2 hours of acute restraint stress, while the other two will remain undisturbed in their cages for the same 2 hours. This keeps the interval between drug administration and behavioral testing the same for all groups. Then all rats will complete a 5 minutes evident past maze test. The maze test has two open arms and two enclosed arms. Rats naturally avoid open elevated space, so more anxious rats will spend less time in the open arms. The dependent variable is the percentage of total test time spent in the open arms, with a lower percentage indicating greater anxiety-like behavior. Finally, I'll analyze the result using a two-way ANOVA to test the mean effect and the interaction. This graph shows my expected results. The no stress plus vehicle group is expected to spend more time in the open arms. Interaction, the lowest anxiety. The no stress fluid group is expected to spend less time, suggests that blocking hippocampus androgen receptors increase anxiety even without stress. The acute stress vehicle group is also expected to show reducing open arm time, reflecting the effect of stress alone. Finally, the acute stress fluid mind group is expected to have the lowest percentage, indicating the highest anxiety. Importantly, fluid mind is expected to increase anxiety more stronger in stressed rats than in non-stressed rats, as shown in the graph. The difference between the vehicle and fluid mind groups is large under acute stress. This pattern will indicate an interaction between stress and treatment. If the expected results are observed, they would suggest that blocking hippocampus androgen receptors may amplify the effect of acute stress on anxiety-like behavior. They would also stress that normal hippocampus androgen receptor signaling might serve about as buffering or preventive role during stress. When this signaling is blocked by fluid mind, that percentage may be reduced, leading to great anxiety after stress. In the terms of this theory, this study could help us understand whether hippocampal androgen receptor signaling generally reduce anxiety-like behavior or whether it is especially important during acute stress. It was exciting Edinger and Frian's findings by showing not only that blocking these receptors increase anxiety-like behavior, but also the size of this effect may depend on stress exposure. This would give us a more precise understanding of the function role. On a particular level, this study may contribute to our understanding of the biological factors that influence vulnerability to anxiety following stress. Although the finding cannot be directly translated into clinical treatment, they may prove an animal model basis for future research on stress-related anxiety. One limited is that I am only use male rats, so the finding may not generalize to females. Future studies could examine whether the same stress depressant effect also occurs in the female rats. Overall, this study will test whether hippocampal androgen receptor blockade interacts with acute restraint stress to increase anxiety-like behavior in male rats. The key prediction is that fragment will reducing open arm time more is in stressed rats than in non-stressed rats. If that pattern holds, it will expose the idea that hippocampal adrenaline receptor signaling may help buffer the behavioral effect of acute stress. Thank you for listening.